So thanks for the intro. And in the next 30 minutes or so, I would like to show you some recent work on how local circuit and cellular, or I'd rather say subcellular mechanisms and architecture in the hippocampus supports the formation and storage of associative memories. So neurosubstrates, as we heard, of course, from Tim's talk, neurosubstrates of learning and memory are studied at multiple scales, ranging from ensemble level representation through single cell responses, such as the place fields of individual place cells, and also at the level of microcircuits of interconnected excitatory and inhibitory neurons, all the way down to the subcellular synaptic and molecular underpinnings of learning and memory. So I think there is an unanimous agreement that subcellular and microcircuit computations play key roles in learning and memory. But the sad reality is that studying them in vivo in the hippocampus is still largely an aspirational goal that is mostly beyond reach. And the reason behind is quite obvious. Even the modern large-scale high-resolution recording approaches, such as multiphoton calcium imaging, but I can also list electrophysiology here, are really powerful when it comes to monitoring single cells or ensemble representations over the course of learning, but they still fall quite short when it comes to accessing subcellular and microcircuit computation. So our favorite approach of in vivo two-photon calcium imaging, for example, provides functional access to a large number of cells, but it does not right away tell us about their local circuit connectivity. So here, for example, the densely packed cell body layer in the hippocampus really complicates the recording, since it's really an asset when it comes to high throughput imaging, but becomes a major problem when it really requires to restrict, for example, optogenetic manipulation to a single neuron or recording from individual dendrites of these cells. So how can we access single cells and their synaptically coupled local microcircuits in the behaving animal? So to overcome some of these limitations, we recently developed bona fide single cell labeling. Yeah, sorry. So single cell labeling methods in combination with optogenetics and subcellular imaging. And the studies that I'd like to show you today really revolve around the method that we call two-photon guided single cell in vivo electroporation. So this is a method that two fantastic postdocs, Tristan Geiler and Adrian Negreant, have established in the lab by basically modifying a single cell electroporation approach that has been previously applied in the neocortex, and they applied it to the hippocampus, to dorsal hippocampus in the mouse. So this procedure allows permanent plasmid-driven expression in individual hippocampal neurons with a controlled combination of transgenes, which are then stably expressed a few days after the electroporation. And these transgenes can be genetically encoded calcium or voltage indicators, factors for monosynaptic tracing, optogenetic actuators, or recombinases to induce targeted gene deletions in single cells. So I will next show you how we use some of these combinatorial approaches recently to start to investigate microcircuit and subcellular substrates of memory. So I could say that the age of the dwarf has come, at least to my lab. So onto the microcircuit question. We know quite a bit about the cellular composition and connectivity structure of microcircuits in each hippocampal subregion. And Many of the postulated computational functions in each subregions are thought to be actually supported by distinct microcircuit connectivity rules. For example, the main input node of the hippocampus in the dentate gyrus, the granule cells, are only connected through feedback or lateral inhibition, which is thought to play a role in pattern separation. And in contrast, the CA3 region is enriched in recurrent excitatory motifs, which is thought to play a role in pattern completion and attractor dynamics. But what about the CA1? So CA1 is the main output node of the hippocampus and the birthplace of place cells. So not only the general role of this region in memory processes is somewhat less well-defined, but all models of CA1 place cells that have been proposed so far, they operate under simplified assumptions that recurrent excitatory connectivity in CA1 is low or non-existent, and the microcircuit architecture is dominated by almost exclusively by lateral inhibition. So the first take-home message that I have for you today is that the local circuit architecture in CA1 is different from the dentate gyrus, and perhaps more similar to the CA3, or maybe between the two, where there are local subnetworks in CA1 that promote feature amplification from a single cell within a subnetwork, but lateral inhibition between subnetworks promotes competitive ensemble selection and sparse coding. And both of these mechanisms can have major implications for the modes and mechanisms of place cell and place cell map formation and dynamics. So let me briefly show you how we arrived at this conclusion. So Tristan sought to use single cell electroporation to simultaneously record the activity of a single pyramidal cells in CA1 together with their local presynaptic interneuron partners during spatial navigation using a monosynaptic retrograde tracing method. So to do this, he expressed calcium indicator in all inhibitory cells that in CA1, and then electroporated a single pyramidal cell with plasmids to express rabies complementation factors, rabies receptor, protein G, a red fluorescent protein, and a green calcium indicator. And then mice were trained to run on a treadmill, and he used a large-scale volumetric 2-photon calcium imaging to image activity of the starter pyramidal cell and all the surrounding local interneurons. And then he injected this tomato-expressing G-deleted rabies virus in the vicinity of the starter cell, following the imaging to prevent any potential toxicity from the rabies during imaging. And after 2 weeks of rabies expression, the identity of each interneuron, such as tomato-expressing presynaptic cells or non-expressing unlabeled interneurons, was mapped retrospectively to their in vivo dynamics. So with this record and tag approach, Tristan now could look at the relationship between the spatial selectivity of a starter pyramidal cell and its local presynaptic partners. And what he found is that in about half of the networks he recorded, the starter pyramidal cell was selective to a location, the specific location on the belt. So it had a place field, as shown here for this example cell, and also in this normalized average tuning curve of this cell centered around the peak of its place field. So when he looked at the activity of the interneurons, he found that the spatial response of the unlabeled cells was relatively high across all positions, and which is consistent with the high baseline firing rate of interneurons that were reported in previous studies. But interestingly, in a number of presynaptic cells, their spatial response showed this marked dip during the traversal of the starter pyramidal cell receptive field, as shown here for these few example presynaptic cells. So, and the level of inhibition between the presynaptic and the unlabeled population was significantly different only at the vicinity of the place field peak. And Tristan analyzed this relationship also using a metrics called in-field selectivity, which reports whether the interneuron activity is higher within the place field of the pyramidal cell, inside versus outside. In this case, it would be closer to 1, or the interneurons are less active inside of the place field than outside, would be closer to negative values. And using this index, indeed, what we found that the presynaptic interneuron population had a more negative in-field selectivity value compared to the unlabeled ensemble. So other experiments that I don't have time to show you actually demonstrated that this place field formation in a pyramidal cell was not a consequence of a spatially localized or even transient reduction of inhibition, raising the possibility that perhaps something completely different happens, such as that the place field formation itself in a single place cell can somehow induce inverse tuning in the presynaptic local inhibitory microcircuit. So to test this, Tristan developed an optogenetic approach to implant individual place cells at control locations by electroporating, as you can see here, a single cell with a redshifted excitatory opsin and a calcium indicator, and the cell responds reliably to short pulses of photostimulation. So, but next he delivered this repeated photostimulation at an arbitrarily chosen location for a few laps. And as you can see here in a subset of session, photostimulation induced a place field that persisted even a few hours after 1 hour in a home cage in the next session, which we call this post session. I'd like to mention here that this optogenetic manipulation approach is very similar to what recently been discovered and described as de novo or electrophysiologically induced place fields by the Maggie lab, and they called it behavior timescale plasticity. So we originally called our method as opto-BTSP, but reviewers oppose this terminology. So I will use the term optogenetic place field induction, or opto-PFI. So with this method, Tristan now again induced a place field in a single cell in the brain and then imaged the surrounding interneurons in the hippocampal area CA1. What you see here is the heat map of the average spatial tuning curves for all interneurons before induction and following successful and failed inductions. And these interneurons are ordered according to this in-field selectivity index around the place, the single pyramidal cell place field. So, and as you can see here, when the induction was successful, a subset of interneurons reconfigured their spatial tuning to develop an inverse anti-tuning negative response around the location of the pyramidal cell place field in this post session. So this is quantified here on the right as a left shift that in the in-field selectivity index of the interneurons following successful but not during failed induction. So together, these results told us that implantation of a single place cell reorganizes interneuron selectivity and induces inverse tuning in a subset of interneurons. But still, then raises the next question: how is it possible that a single cell can have such a robust effect on interneurons that leads to reorganization in the inhibitory network? So to address this, Tristan induce the place field in a single pyramidal cell and image the activity of all the surrounding other pyramidal cells. So as you can see here in this single example and then the summary data, the stimulation of a single pyramidal cell actually was able to increase the activity that in a subset of surrounding other CA1 pyramidal cells, which is consistent with previous reports of sparse recurrent excitatory connectivity between CA1 cells. Well, what happened next, he induced the place field in the single starter seed cell and see what happens in the surrounding other pyramidal neurons. And following successful induction, the pyramidal cells, coactive pyramidal cells, formed place fields, as fraction of them. And the distribution of this newly formed place field was enriched near the location where the original seed cell was induced, as it's shown here on the heat map, the cumulative distributions. And again, this happened only when the induction was successful. So putting this together, these results are consistent with the subnetwork structure in CA1, where there is a coordinated connectivity and plasticity between a subset of coactive pyramidal cells and their associated inhibitory subnetworks that allows single cell spatial tuning or feature tuning to be scaled from a single cell to multicellular assemblies. So this raises the next question: how these subnetworks actually interact. And we investigated this in another set of experiments led by former graduate student Sebi Rolotti and two postdocs, Mohsin Ahmed and Miklos Szaboszlai in the lab. I don't have time to show you the full study. I just give you the relevant punchlines. So first, what we found is that when we increase the number of the photostimulated pyramidal cells, and in this case, pyramidal cells that were randomly allocated across subnetworks, the magnitude of the lateral inhibition scaled with the increased coactivation of pyramidal cells. And second, we found that when we try to induce now place fields at the ensemble level, not at the single cell level, but in these sparsely labeled pyramidal cells, the efficacy of the induction depended on the density of the coactivated pyramidal cells in a way that ensemble PFI, ensemble opto-PFI fails at medium densities about 5 to 10% of labeled pyramidal cells, that as indicated and quantified here as a lack of shift of the place fields towards the stimulation zone. So these results together consistent with the model that these subnetworks in CA1, they interact mainly through lateral inhibition, and this lateral inhibition constrains the size of the ensemble that can be new ensemble that can be formed. So there were other experiments showing that a temporal relief of local inhibition actually allowed us to induce place fields in larger ensembles, further supporting the role of lateral inhibition in regulating place field formation. So I don't have time to show those experiments. That's just the final set where I'd like to show you here that when we induce place fields at the ensemble level, these de novo synthetic place fields will have actually a behavioral effect. And to do this, we sparsely labeled the subpopulation of pyramidal cells across both hemispheres with the redshifted opsin, and also introduced inhibitory opsin to interneurons in CA1. And then we did bilateral photostimulation to coactivate pyramidal cells whilst we suppressed interneurons in this goal-directed learning task, where the animal has to learn the location of an unmarked operantly delivered water reward, and then this reward zone is then moved to a new location over subsequent sessions. And the trick was that while the animals were actually learning the first reward zone, we applied this ensemble level opto-PFI at a randomly chosen location on the belt. And during the next session, the reward zone was moved to this very location, so we could see whether this ensemble opto-PFI had behavior effect during the learning. And briefly, what we found is that across various learning conditions and controls, only those animals that had an enhanced pyramidal cell stimulation together with optogenetic suppression of inhibition during the opto-PFI showed faster learning and increased anticipatory clicking rate near the reward zone, which indicates enhanced learning performance. So this is perhaps one of the first demonstrations, causal demonstration that place cells and place cell map, they have role in spatial learning. So I summarize this first part very quickly. What we found is that the CA1 cells do not operate in isolation. Rather, CA1 is enriched, more enriched in connectivity motifs, excitatory connectivity motifs that we previously thought. And there is a coordinated activity and plasticity between pyramidal cells and interneurons that allows this scaling of feature selectivity from single cell to ensembles within a subnetworks, but between subnetworks, there is a robust lateral inhibition that limits the size of the new ensemble that can be formed, and perhaps provide a mechanism for competitive ensemble selection. And perhaps these two together are optimized for optimal coding in CA1. And finally, we can propose that there is a new plasticity mechanism, or plasticity inducing approach rather, that requires both increased excitation and suppressed inhibition. And this can be together used to generate synthetic ensembles and manipulate and control adult behavior. So just very briefly, I'd like to tell you that, of course, there's many unknown here, and including how this initial connectivity structure is set up, and whether this is development or experience-dependent plasticity. So we don't know the answer. I leave you one teaser here, that we can detect this ensemble structure, subnetwork structure, before the light touches the brain. So we find that these co-active neurons, co-recruited neurons, they have higher pairwise correlation during rest and closer place fields during exploration. But what's quite intriguing is that we also find that this like-to-like relationship among these recruited neurons is actually more pronounced for cells that are closer to each other, with about 100 and 150 microns. So to me, that indicates that perhaps there is a developmental connectivity scaffold organizing subnetwork structure. So in the remaining 10 minutes or so, I move to different scales, zoom in even more and talk about dendrites very briefly. And why they are interesting, because of course, as you know, they receive all the excitatory and vast majority of the inhibitory cells in the complex dendritic fiber of pyramidal cells. And in the simplest case, these inputs come to the distal and proximal dendrites, and they are summed linearly before a axonal axosomatic threshold is applied as in the form of somatic output. But what is important that these complex dendrites, they are equipped with a high variety of voltage-gated conductances that support a high level of nonlinear computations, including active backpropagation of the somatic action potential or local spike generation out in the dendrites. And these local spikes or backpropagating signals are both really powerful associative signals for synaptic and intrinsic plasticity. So we know that, and we know a lot about these processes in vitro, including this ever-growing catalog of anatomical connectivity. However, the in vivo activity dynamics of dendrites in the hippocampus has remained so far almost completely unknown and uncharacterized, with only 2 studies with dendritic calcium imaging in a awake animal. And in particular, the relationship between dendritic activity and learning-related oscillatory dynamics remain unknown. And what do I mean by this? As you all know, there is a strong temporal component of hippocampal memory operations that manifest in a form of 2 antagonistic network oscillations, such that memory encoding is often associated with, as I showed you, the place cells, the place fields, 5 to 10 hertz rhythmic oscillations, during which the place cell firing advances relative to the theta phase. This is called phase precession. And in contrast, memory consolidation, thought to be primarily associated with high-frequency shortwave ripple oscillations, which occur during rest and immobility, during which these place cell sequences are replayed in a time-compressed manner. And these 2 stages together, they often called and referred as a dual process model of memory, with theta encoding and ripple-associated consolidation. So the dendritic activity during these oscillatory states is completely unknown. And an added complication is that calcium has limited sensitivity and resolution to relate dendritic electrical signaling to network oscillations. And recently, voltage indicators came on board to record fast electrical signals, and that's what we used in another study to record dendritic activity across the entire somatodendritic surface of C1 pyramidal cells, which was led by a fantastic postdoc, Adrian Negrean, in the lab. So Adrian used this electroporation method to express a green fluorescent voltage sensor ASAP3B developed by Michael Lin's lab, together with an RFP and in CA1 cells, and after the plasmid expression, in single cells, we use the fast sequential imaging of multiple regions of interest over the entire somatodendritic surface during bouts of head-fixed navigation and rest, together with simultaneous LFP recordings. So I show you here an example cell. So recordings here covered the soma, the basal dendrites, the apical trunk, the radial obliques, all the way to the distal tuft of these pyramidal cells. And Adrian managed to detect depolarizing events across the whole somatodendritic surface during locomotion and periods of rest. So he carried out detailed reconstruction of all the cells to relate the position of the region of interest to the electrical signals. And he indeed, just briefly, he found that the average depolarizing event waveform was quite similar between recording locations. Three punchlines I'd like to give you with this fast voltage imaging. The first one is that, so this fast imaging methods allowed us to relate dendritic electrical signals in the subthreshold and suprathreshold regime to extracellular theta oscillations during locomotion. So when Adrian mapped the theta filtered membrane potential oscillation phase on the morphological reconstruction, as illustrated here, he found that a prominent phase gradient that across the dendritic tree as a function of the somatic path distance of the recorded region of interest. He also found that the amplitude of the subthreshold oscillations increased from the soma to the tuft dendrites. And he finally also found that the suprathreshold depolarizing events, dendritic spikes, for example, they tend to occur at the peak of the local oscillation, not at the peak of the somatic oscillation, so their phase also shifted with distance. All in all, what it tells us that indeed, theta oscillations are manifested and organized across the dendritic tree as a traveling wave. And just very briefly, I mentioned that during shortwave ripples, the picture was very different. Across all the dendritic compartments, we found a prominent hyperpolarization in the subthreshold membrane potential and the suppressed depolarizing event rate. The third component, third punchline I'd like to give you is the feature selectivity of dendritic events during head-fixed navigation that we looked at. And we indeed found that a subset of the dendritic segments exhibited significant spatial tuning, as illustrated here, the heat map of the normalized depolarizing event tuning curves, and they are sorted their peak position along the treadmill. And as you can see here, these dendritic branches, they tile the space. And when we plotted and clustered these tuning curve distances, the matrix showed some level of co-tuned blocks, but the majority of the segments were not co-tuned, including the cell body, suggesting that the dendritic arbor is compartmentalized internally as well as compared to the soma. So voltage imaging and calcium imaging studies that I don't have time to show you indicated that really dendritic branches have an unexpected degree of heterogeneity in their feature tuning that's largely independent from the soma, which raises the question, what's the cellular molecular mechanism that regulates this heterogeneity and spatial tuning preference in dendrites? So in the last 5 slides or so, I'd just like to briefly walk you through a recent study that we finished in collaboration with with my fantastic colleague at Columbia, Frank Poulot, spearheaded by great postdoc Justin O'Hare, in which we identified at least one mechanism, intracellular calcium release, that regulates compartment-specific tuning in dendrites. So intracellular calcium release, as you know, occurs through IP3 or ryanodine receptor-mediated mechanism in response to strong synaptic stimulation. And the cytosolic calcium is then buffered by mitochondria, which controls the level of cytosolic calcium. And a few years ago, Frank's group identified PDZD8, a tethering protein that brings the ER and mitochondria together and enables mitochondria to buffer the calcium in the cytosol. So once we delete, and when they deleted PDZD8 gene, it increased the cytosolic calcium level in response to strong synaptic stimulation, so which provided us with the gain of function manipulation to increase cytosolic calcium levels in response to synaptic activation, which is, as you know, key signaling component for neural activity. And what Justin did, he used our favorite approach I show you a few times, single cell electroporation, to electroporate criticombinase in a conditional FF animal, and then to generate single cell that knockouts for PDZD8. It also did it in the in Y-type animals in combination with calcium sensor and optogenetic actuators. So, and he imaged the basal and apical dendrites across the tree. So it takes a lot, but we are almost there. 2 more slides. So when Justin imaged the activity of multiple dendritic branches of basal and apical and basal dendrites in Y-type and knockout cells, and he calculated the tuning curve of the dendrites and the soma, and he calculated the place fields of the dendrites and their somatic place field, and calculated then the distance, the minimum circular distance on the belt between the somatic and dendritic place fields. And what he found is that with augmented intracellular calcium release, the augmented release was associated with a significant shift in the dendritic place fields towards their soma, as indicated here by the lower values of the soma dendrites place field circular distance. He didn't find this difference, that augmented ICR did not affect the basal at the dendritic tuning. So together, these results identifies ICR as one mechanism that controls dendritic feature selectivity. And perhaps just as interestingly, what we find is that this is compartment specific. It's more pronounced for the apical dendrites compared to the basals. So really, I stop here, and I just mentioned that Justin identified other main differences of ICR manipulation on somatic outputs or broadening BTSP-like induction kernel or stability of the place fields. What I'd like to do is just very quickly recap the second part on dendrites. So what I first showed you that branch level dendritic compartments have a high degree of functional autonomy from the soma, both in their intracellular oscillations and spatial tuning. We identified the one mechanism that regulates this dendritic feature selectivity and shapes also output level somatic place fields. And why is it interesting? It's potentially interesting because these studies that we find a prominent intrinsic oscillation in dendrites, it gives credence to insofar computational models of oscillatory or soma dendrites interference in generating hippocampal temporal codes such as phase precession. We hypothesized that also hippocampal pyramidal cells, similar to their neocortical cousins, they can acquire any output level somatic tuning by potentiating the right combination of their dendrites. So all in all, these findings supports the notion that dendritic compartments, and in particular branch level compartments, are the fundamental syntactic computational units in pyramidal cells during memory operations. And here we are. I'd like to thank everyone for their in the lab for their fantastic work, and all the collaborators inside, outside Columbia, and also funding sources. And I'm happy to take questions.